a chapter which is nucleic acid, and we will discuss about the structure of nucleic acid. Also we are going to talk about the structure of DNA and RNA. This particular chapter is relevant both for class 11 and also for class 12. So let us begin. Okay. So let us understand what are nucleic acids. So nucleic acids are one of the most fundamental biomolecules. Those are present in all living organisms. Okay. So when I say biomolecules, biomolecules are broadly of two types. One is macromolecules, which are of larger molecular size, more than 1000 Dalton, and there are micromolecules which are less than 1000 Dalton. Okay. And nucleic acids are the biomolecules which are more than 1000 Dalton, and hence they are called as macromolecules. Okay. And these nucleic acids are the polymers, and they are polymers of what? Like for example, we know that proteins are the polymers of nucleic acid. Polysaccharides, they are the polymers of monosaccharides. Similarly, nucleic acids are also called as the polymers. So when I say polymers, so they should be made up of several monomers. Okay. So they are made up of several monomers which are called as nucleotides. So for example, this is one nucleotide, they will link with another nucleotide, which will link with another nucleotide like this, and this is what is going to make a polynucleotide. And nucleic acids are nothing but the polynucleotide chain. Okay. We have two different kind of polynucleotides which exist in the living organisms. One is DNA and another is RNA. So DNA is a polymer of two polynucleotide chains. Okay. But there are DNAs which are present which consist of single polynucleotide chain, which we will discuss as we go along with this lecture. But there exists also RNA. RNA is most commonly single stranded. So this consists of only one polynucleotide chain. So when we are talking of nucleotides, which are the monomeric units, the nucleotides consist of three different groups, subgroups. Okay. One of them is a sugar molecule, and this sugar molecule is a pentose sugar. So when we talk about pentose sugar, this is a 5-carbon sugar. Okay. It also, in addition to that, it also have a nitrogen base and a phosphate group. The phosphate group determines the charge on that molecule. Okay. DNA and RNA are acidic in nature, and that acidic in nature comes due to the presence of the phosphate group. Okay. Let us look at the discovery of the nucleic acids. Nucleic acids were discovered way back in 1869 and by a Swiss physician whose name was Friedrich Miescher. So Friedrich Miescher was a medical doctor by profession, and he was studying about the lymphoid cells, and extracting the lymphoid cells is slightly difficult. Okay. And hence he chose a very important pathway of isolating them from the pus. So pus is the kind of extracts that we get from the wound, and pus is extremely rich in WBC, and we know that WBCs are the nucleated cells. Unlike the RBCs, which are anucleated, WBCs or white blood cells are nucleated cells, and therefore he isolated the nucleus from these cells and wanted to study them. And he found that he analyzed the components of all the components which are present inside the nucleus, and he termed these components as nuclein. Okay. So nuclein means the components which are present inside nucleus. Okay. So this part stands for nucleus, and IN stands for inside. Okay. That means the components which are present inside the nucleus. And later on, as these molecules are acidic in nature due to the presence of phosphate group, these are called as nucleic acids. Okay. And this terminology was given by Altman in 1889. Okay. If we will move to study the structure of the nucleic acid in a greater detail, as I was highlighting earlier. So, this consists of 3 different components, which is a pentose sugar, a nitrogen base and a phosphate group. So, let us look at them individually. What is the structure of each of the molecules look like? So, this is what is the pentose sugar. This is a 5 carbon sugar, that is why it is called as pentose. Glucose consists of a 6 carbon sugar, that is why it is called as hexose. So, as we can, so this sugar can be present both in the cyclic form. So, this is the cyclic form of the sugar and this is the straight chain form of sugar. And this is how the numbering is done. Okay, it starts with 1 prime, okay, 1, 2, 3, 4 and 5. Okay. So, this is how the numbering of the sugar is done. If we look at the sugars, those are present in DNA and RNA, they are slightly different from each other. Okay. How they are different from each other? So, just look at the structure of the sugar which is present in RNA. That is why this sugar is called as ribose sugar. The full form of RNA is known as ribonucleic acid. Okay, that means it has the sugar ribose. If we look at the sugar ribose at the 2 prime position, okay, this group that is present here is a OH group. Okay, but if we look at deoxyribose at the 2 prime position, if we look at one of the O group is missing here, okay, and only H group is present here, and that is why it is called as deoxy. Deoxy means deficient in one oxygen atom. Okay, so that is why this is called as deoxyribose sugar, and this one is called as ribose sugar. As we know that hydroxyl groups are one of the most reactive molecules, okay, or the OH group is one of the most reactive molecules. As OH group are the most reactive molecules, they can participate in several reactions, and that is why as ribose sugar is a part of RNA, RNA is a most unstable molecule because they can participate in several biochemical reactions, whereas DNA is a more stable molecule. That is why DNA is the genetic material in most of the advanced group of organisms, whereas RNA is the genetic material in most of the viruses because it is still a primitive form of organism. And that is the most reason, you have to remember this thing, that RNA viruses are more prone to mutations due to the presence of this hydroxyl group, as they are very unstable in nature. Okay. So, this is what fundamentally the structure of the ribose sugar is. Okay. So, let us now move ahead to the next component of the nucleotide, which is the nitrogen base. This nitrogen base is the most significant part of the nucleotides. Okay. There are different types of nitrogenous bases present in DNA and RNA. Okay. And how many of them? In DNA there are present 4 different types of nucleotides, nitrogen bases. In RNA also present 4 different types of nitrogen bases. They are slightly different from each other. But if we broadly categorize the nitrogen bases, they can be categorized into 2 types. One of them is called as the purine, the other group is called as the pyrimidine. Okay. If we look at pyrimidine, the pyrimidine is a single ring structure. Okay, as you can see here, this is a single ring structure, whereas the purines are a double ring structure. The name is slightly smaller, purine, so the larger is the structure. This is how you need to remember it. Okay. So, purines are the smaller name but a larger structure, whereas pyrimidines are a larger name but a smaller structure. Okay. And if you look at the purines have 2 heterocyclic ring, okay, because this is 6 number of molecules here and there are 3 molecules over here. So, this is a heterocyclic ring. Okay. Whereas here it is only 1 ring. Okay. So, there are total 5 different types of nitrogenous bases present. What are the pyrimidines present? The pyrimidines which are present in the living system are cytosine, uracil and thiamine. uracil is present in RNA in place of thymine. So, thymine is not present in RNA, it is only found in DNA, whereas uracil is only found in RNA, but not in DNA. But the adenine and guanine are fixed. So, they are present both in DNA and RNA. So, purines are fixed for both of them, whereas the pyrimidines vary. Okay? So, let us look at the type of nitrogen bases which are present in DNA as well as RNA. If you look at the purines, it is constant. Okay? Both contains adenine and guanine. Okay? Whereas in DNA, cytosine and thymine is present, and in RNA, cytosine and uracil is present. That means thymine is replaced by uracil in case of RNA. Okay. So, this was a question that came in NEET 2019. Purines found both in DNA and RNA. So, it is asking that what are the purines that is present both in DNA and RNA. We know that purines are common both for DNA as well as RNA. They are not different, they are same, and those are what? Adenine and guanine. So, therefore, answer is going to be the third option. Adenine and guanine are the purines. Okay. So, now let us look at the next group which determines the charge on the DNA molecule or the RNA molecule is the phosphate group. Okay. The phosphate group which is present is called as the phosphoric acid, H3PO4. Okay. And the phosphoric acid is attached with the sugar molecule. Okay. If you look at here at the 5 prime position, the molecule which was present CH2OH. Okay. This is a hydroxyl group that was present. Okay. And the hydroxyl group of the sugar is reacting with the hydroxyl group of the acid molecule over here. Okay. So, as we know that the combination of an acid and an alcohol results into an ester. So, therefore, the bond that is getting formed here is an ester bond. Okay. But as phosphorus group is linked over here, such kind of ester is called as a phosphoester bond. Okay. Please remember this thing, because this is going to be very, very critical, because here at the 5 prime position the phosphate group is linked with a ester bond, and therefore, this kind of bond is called as phosphoester bond. Okay. So, this will be a very important question that can come in the exam, that at the 5 prime end the phosphate group is linked by which bond? That would be phosphoester bond. Okay. Let us now move on. So, we know that the pentose sugar, how the phosphate group is already linked, that is what we have already discussed about. Okay. Now, let us see how the nitrogenous base is linked with the pentose sugar. Okay. And that is what is going to make the complete nucleotide molecule. Okay. When the nitrogen base is going to be linked with the pentose sugar, they can be either linked at the N9 position, and that is in case of the purine, or in case of the N1 position of pyrimidine. Okay. So, here it is linked with the N9 position. Okay. Okay. And here it is the C1. So, the linkage is between C1 to N1, and the bond that is produced is called as a glycosidic bond. So, glycosidic bond is a linkage of a sugar molecule with any other molecule. The glyco part stands here for sugar, right? And the linkage which help in connecting the sugar with any other molecule is called as a glycosidic linkage. Okay? That is what is a glycosidic linkage, which is a condensation reaction again. Okay? As we know that when the pentose sugar is going to be linked with the nitrogenous base, the molecule which is going to be produced is called as a nucleoside. Nucleoside. So, what is a nucleoside? Nucleoside is a combination of pentose sugar plus nitrogen base. So, that is what is a nucleoside. Okay. But when the nucleoside is going to be connected with a phosphate group, then it would be called as a nucleotide. When all the three molecules are present together, then it is called as a nucleotide. So, nucleotide would be pentose sugar plus nitrogen base plus phosphate group. Okay. So, this is what is going to make the nucleotide. So, if we only talk about a nucleoside, for example, when adenine as a purine is connected with a pentose sugar, then it is a nucleoside, and the kind of nucleoside it is going to make, it will be called as adenosine. Okay, adenosine. And when guanine, okay, will be connected with a pentose sugar, it will be called as guanosine. But when this pentose sugar is a deoxyribose sugar, then it will be called as deoxyadenosine. And when the pentose sugar is deoxyribose and this molecule is guanine, it will be called as deoxyguanosine. Similarly, the other molecules are given nomenclature, could be named accordingly. Okay. But when three of them are connected with each other, all the three, adenine molecule, pentose sugar as well as the phosphate group, the molecule which is going to be produced is a nucleotide, and what we will call that? Adenosine monophosphate. Adenosine monophosphate, because only one phosphate group is linked over here. That is why this molecule is called as adenosine monophosphate. If there would be another phosphate group would be linked here, that would be called as adenosine diphosphate. Then if another group, three phosphate groups, that would be called as adenosine triphosphate. The ATP, which is called as the energy coins of the cell, okay, are nothing but a nucleotides, because they are exactly the same like the components of a DNA molecule. Okay. So, let us now look at the examples of some pyrimidine based nucleotides as well as purine based nucleotides. Okay. If we will talk about the pyrimidine based nucleotides, there are three different pyrimidines which exist both in combination DNA and RNA. For example, if it is linked with the cytosine, then the nucleotide which is going to be formed is called as cytidine monophosphate. If it is linked with uracil, then it will be called as uridine monophosphate. Okay. And if it is linked with thymine, it will be called as thymidine monophosphate. Okay. And if it is a DNA molecule or a deoxyribose molecule, then it will be called as deoxy, okay, deoxythymidine. And the linkage that is happening here with the sugar molecule is a C1N1 glycosidic bond. That is what you have to remember very carefully. With the pyrimidine molecule, the linkage, the glycosidic linkage that exists is a C1N1 linkage, because the 1' carbon here is linked with the N1 position of the pyrimidine. Whereas, in case of purine, the linkage is with the N9 position and the C1 position here. So, therefore, this is called as C1N1 glycosidic linkage. Okay. So, now let us look at, we have already discussed about the structure of one of the nucleotides, but we know that nucleic acids are polynucleotides. That means more than one nucleotide they join with each other. So, let us now discuss how these nucleotides join with each other, and this is the critical part to understand. For example, this is one of the nucleotides here and there is another nucleotide here. Okay. So, these are two different nucleotides, and this is the phosphate group which is present in one of the nucleotide molecule. Okay. So, this is one of the phosphate group present over here, okay, this one. and this is the hydroxyl group which is present on the phosphate molecule, phosphate group, and this is the hydroxyl group which is present at the 3' position of the nucleotide number 1. So, this is nucleotide number 1 and this is the nucleotide number 2. Okay. So, what will happen? The phosphate group here of the nucleotide number 2 is going to again react with the hydroxyl group of nucleotide number 1 at the 3' position. So, there would be release of a water molecule with the combination of H and OH over here, okay, and then it will form another ester bond over here. So, the molecule which is going to be formed will look like this. So, this is a ester bond getting formed here and another ester bond is already formed here. Okay. So, there are now 2 ester bonds which are formed during the linkage of one nucleotide with another nucleotide, and therefore, the bond that is getting formed is called as a phosphodiester linkage. Okay. And this is a condensation reaction because the molecule is getting condensed with the release of a water molecule. Okay. So, therefore, what we can say that a phosphodiester bond occurs because this phosphodiester bond is getting formed, okay, when exactly 2 of the hydroxyl groups, suppose this is one of the hydroxyl groups of the H3PO4 phosphoric acid and this is the other hydroxyl group of the phosphoric acid. So, when both of the hydroxyl groups of phosphoric acid reacts with the hydroxyl groups on the other molecule of the sugar, and therefore, it generates 2 ester bonds which is therefore called as a phosphodiester bond. Okay. So, a phosphodiester bond is produced when one nucleotide is linked with another nucleotide, but only a phosphoester bond is produced when the sugar is linked to the phosphate group. Understand the difference between phosphoester and phosphodiester. Phosphoester is produced when the phosphate group is linked with the 5' position of the carbon atom, okay, of the pentose sugar. But a phosphodiester is produced when 2 nucleotides are joined with each other. Okay. So, let us now look at this question. Okay. 2 free ribonucleotide units are interlinked with what? Okay. So, the options which are over here is a peptide bond, covalent bond, hydrogen bond, or phosphodiester bond. Okay. If we look at the options that are given here, so when you are talking about ribonucleotide, so ribonucleotide means these are the nucleotides which are present in RNA. Okay. And we just discussed that nucleotides are linked with each other by phosphodiester bond. So, the more appropriate answer for this one is going to be phosphodiester bond. Okay. So, now let us, we have already discussed about how the polynucleotides are produced, and polynucleotides are the DNA or the RNA, okay, are the genetic material of most of the organisms. Okay. DNA is the genetic material in both prokaryotes as well as eukaryotes, whereas RNA is the genetic material in some of the viruses. Okay. If we look at the types of DNA that exist in nature, there are broadly 2 classes of DNA depending on their shape, how they are arranged inside the cell. One of the type of DNA which is present in the prokaryotic cells are called as the circular DNA. Okay. So, prokaryotic cells have a single DNA and that DNA is a circular DNA, whereas the eukaryotes, the DNA is not circular over there, it is linear. So, that is the major difference between the prokaryotic DNA to that of the eukaryotic DNA. Also, if we look at the type of DNA that exists in the organisms, okay, so in some of the viruses, the DNA is single stranded, it is not double stranded, okay, and such kind of DNA is found in 5X174. Okay, it is a single stranded virus, okay, DNA virus, and in all other organisms, okay, for example, in bacteria or in eukaryotes, DNAs are double stranded. Okay. So, there exists also single stranded DNA and also double stranded DNA. So, let us look at the structure of DNA which was kind of elucidated or proposed by Watson and Crick in 1953. Okay. What this model states that, okay, this is the B form of DNA. So, B form of DNA is the most prominent form of DNA. There exist 3 different kind of DNA which we are going to discuss in another lecture. So, B form of the DNA is the most important form of DNA that exists in the nature. Okay. And if we look at the structure of the B form of DNA, so the DNA consists of 2 polynucleotide chains, as you can see here. So, one chain is here, the other chain is over here. So, there are 2 polynucleotide chains, and these 2 polynucleotide chains, they run around each other, they are coiled around each other, okay, in a helical manner. They are forming an helix, okay, and they are anti-parallel in nature. If you look at the direction here, so this one starts from 5 prime to 3 prime here, and the other chain starts from 5 prime to 3 prime here. Okay. That means both the chains are anti-parallel in nature. If one chain runs in one direction, the other chain runs exactly in the opposite direction. Okay. So, these 2 chains are anti-parallel and twisted around each other. Okay. If we look at the structure of the DNA, it is kind of twisted around each other, and when it takes each turn, okay, suppose this is a complete turn, okay, when the DNA takes a complete turn, the distance that it covers is around 3.4 nanometer, and if we convert that value in angstrom, this is going to be 34 angstrom. The question can come both in the form of nanometer as well as angstrom. Okay. So, therefore, remember both the values. Okay. And the width between the 2 strands is around 2 nanometer or 20 angstrom. Okay. The distance which is covered by the one turn is called as a pitch. So, the pitch of DNA is going to be 3.4 nanometer or 34 angstrom. And in each turn, the DNA has around 10 base pairs. Okay. It has 10 base pairs in each turn. Okay. So, therefore, when it goes one base pair, okay, which is called as a rise, okay, because it is just like one step of the staircase. Okay. When you take one step of the staircase, which is called as the rise, and if there are total 10 rises in one turn, that we know already. So, if we divide 3.4 divided by 10, so this distance that is going to come is 0.34 nanometer. Okay. If we convert that into angstrom, that is going to be 3.4 angstrom. Okay. So, this is what I was talking about, the polarity of the DNA. If you look at structurally here, if suppose this is the strand 1 and this is the strand 2, okay. If you look at here, the strand 1 runs from this position to this position. Always it is read as 5 prime to 3 prime, okay. So, 5 prime means where the phosphate group is linked, okay. And here it is the phosphate group is linked with the 5 prime position, which is also 5 prime to 3 prime. So, therefore, the 2 chains are anti-parallel in nature, and the 2 chains are held together by the hydrogen bonds between the nitrogen bases. Okay? Because if you see here, the nitrogen bases are projected from the sugar phosphate backbone. Okay? Because if you look at here in the previous slide, this particular chain is the sugar phosphate backbone, and nitrogen bases they have arisen from this sugar phosphate backbone. So, they arise from the sugar phosphate backbone at an angle of 90 degrees. Okay? And also from the opposite strand, there will be another sugar phosphate backbone. So, there will be another nitrogen base, and these nitrogen bases always will pair with each other. Okay? And the pairing is always a complementary pairing. Please remember this thing. Adenine, which is a purine, is always going to do complementary base pairing with a pyrimidine, and that is thiamine. Okay? And guanine, which is a purine, going to do complementary base pairing with another pyrimidine, which is cytosine. So, G will pair with C and A is going to pair with T. and the number of hydrogen bonds those are going to be produced between A and T is 2 hydrogen bonds, and the hydrogen bonds that is going to be produced between G and C is 3 hydrogen bonds. Okay. So, the number of hydrogen bonds produced here is much higher. So, therefore, DNA containing more GC residues, more number of GC will be a stronger DNA as compared to a DNA consisting of only 80 residues. Okay. So, this is what structurally it looks like. So, there are 2 hydrogen bonds here and there are 3 hydrogen bonds here. So, let us now discuss some of the questions related to this. 1 turn of the helix in a B form of DNA is approximately how much? Okay. So, we are talking about the complete turn, which is the pitch, okay, and the pitch is how much? We have already discussed 3.4 nanometer or 34 angstrom. Okay. So, the answer therefore is going to be this one. Antiparallel strands of DNA molecule means that one strand turns anticlockwise. The phosphate groups of the DNA strands at their ends share the same position, and the phosphate groups at the start of the 2 DNA strands are in opposite position, and one strand turns clockwise. We know that the starting position is the phosphate group is the 5 prime position. Okay. And we have seen that in a DNA they are antiparallel. The 5 prime position is present in opposite strands. Okay. So, therefore, the phosphate groups are present at the start of the DNA in the opposite positions, opposite poles. Okay. So, therefore, this is going to be the correct option for this one. We will discuss more on this chapter in the coming session.